Hemant Sarin earned his degree from University of the Highest Honor since 2014. He has been studied in small molecular and the structure functions relationship, cell definition, and the topic of today's meeting is cancer, genomics, and biomarker. Thank
Yes,
you.
I appreciate the introduction. So yeah, my talk is on personalized the personalized determinations of toxicity related carcinogenesis in the example of what we call environmental polyhalogenates. Uh, these are high risk ecologic risk through the chain of organisms. They've accumulated, and this can be the various, and this is by depth, depth by distance from the land-ocean interface. There's been, and this is uh, uh, or they can be superficially located in waters secondary to this to this most commonly plastics that we use, everyday plastics that have that are buoyant and can leach over time superficially to as far as in deeper waters, deep sedimentation. There's sedimentation of the more dense molecules. These are mainly these are halogenates, halogen substituted molecules with heavier atoms, and the majority of sources are PCBs are from industrial products, and the bisphenols, another class, are in everyday use plastics. The polychlorinated biphenyls, they're benzyl rings with delocalized pi bonding, carbon bonding, and they're used. They're actually the non-conductive, the dielectric materials. They're in all sorts of capacitors for, to separate the charge, separate charge, and they have an optimal dielectric constant as compared to in vacuum or air and air. Um, they're or air, and they've accumulated um, uh, um, uh, vice over time. And then the bisphenols that are in these in plastics with polyvinyl chlorides, and these are hydroxylated compounds with halogenation, and they interlock non-covalently to make plastics what they are. Uh, the primary there's several different several different there's subcategories of these TCD p dioxin tetrachlorobenzodioxin. This has been shown to be a IR group 1 carcinogen from the International Agency for Research on Cancer, and it initially was determined intracellularly, to be localized intracellularly by in vitro in vitro in cell cultures, um, and concentration when applied in that, and this is initial work that then subsequently the pathway has been extensively researched since the 80s, um, and these are the pathway involve and that's since the advent of EMSA, and then immunoprecipitation, immunoprecipitation, um, that antibody based chip assays, and then computational genomics that has being able to predict certain sites, response elements, where as to where the receptor binds and its co-receptor adapter aren't the aerial related, also known as the alternate subunit of hypoxia-inducible factor, as to where they could these what sequences they can bind to, and then further confirmed by DNA microarray for expression, as to in terms of differential recruitment. And this has led to more. This has led to uh, uh, the identification of various enzymes, target genes, phase one enzymes that do the initial hydroxylation and for the subsequent phase two detoxification by the activation of the NRF2 pathway and the ether-like pathway during de-repression by NRF1, and this is the work that was in now the flox deletion constructs with albumin detection, um, as to the role of this repressor NRF1 for this pathway um, when it is inactive, and it's decreased when it becomes active. All in all, there's a causal association with biliary carcinogenesis and also hepatobiliary. Uh, Other molecules in this subclass, a subclass of molecules in polyhal are the secondary PCBs. These are not actually PCBs, coplanar PCB type PCBs, and these are less toxic. Can be compared to TCD as far as toxicity. They're similar, but less, somewhat less. The important thing is that they have, although as far as this pathway is concerned, compared to TCD, this is an estrogen receptor positive pathway as compared to the form, as compared to the former, uh, and in which metabolic pathway effects are noted by increase in transcription of genes that are involved in these in such pathway. As far as one pathway that's overexpressed, these are lower risk for TCD-like effects, but have other effects. This is what has been shown in recent work by in the research that I'm presenting on from the uh, um, uh, and this compares to what I'm calling here non-classic PCBs. These are ones that are orthoplanar now, orthosubstituted, um, and they have different properties. Their equivalency factor is significantly less, on the order of 10 orders to 11 orders less than DCD dioxin, but they're toxic for another reason. And in terms of apoptosis being more the pathway resulting in de-differentiation of cells, resulting during which there's differentiation of cells. Estrogen, this is an estrogen B beta positive pathway, or close to a one that is positive, and I will discuss this in a few slides. This is an all and there's all the gene expression is different. Um, uh, Now, in terms of what the more recent the research that I'm discussing is is, um, is that this builds on research from that was published in 2016 on that actually gene transcription can be the conserved mechanism underlying it is horizontal establishment of the horizontal reading frame, uh, just prior to, as these as transcription factors that regulate the process specific to genes attached to their are attaching to various sites around the gene, the promoter and enhancing sites, which may be or may not be occupied by a ligand. Yet these genes are responsive to changes in transcription um, uh, modeled, which has been modeled uh, in the cell as it being effective pressure that results in this. Now, in terms of and it is also and the gene expression results from the duration at this at the p effective for resulting in gene transcription, which can be used to measure cell compliance. Although classically, historically, atomic force microscopy has been used to detect the compliance directly, and it's actually the techniques that are used involve aspiration of cell membrane to detect this the compliance and determining of the Young's modulus. Um, uh, the method that I'm going to be that has been applied in this more recent research is that the intergene spaces, intergene distances are measured, and these are based on the genome map where genes are exactly located, um, uh, and then the relative tropy, the tropy between in terms of the balance, in terms of the counts are determined in a from an upstream to downstream direction, depending on the forward or reverse orientation of strands. And there's a sequential summation method where sub-episode blocks are established, and they're integrated from an initial constant to a variable depending on the gene locus and the actual the gene length. And this results in this quotient, which is an indicator of effective pressure that would be required to make the DNA horizontal for it to be transcribed. There's a validation to this method, and there's also it is a and it's been shown that and it is this results in linearization of the anisotropy, and there's also a second stage linearization process, which is research in progress for in terms of more detail on what has been what has been shown. And one can predict the angulation in terms of a gene tropy, a loop with respect to that gene, in terms of positioning, and a model can be developed in terms of has been developed and can be furthered. Um, uh, um, my screen. Okay, there it is. Now, TCD associated gene expression. So there's several standard marker genes in this more recent research, and this is it's been initially considered, and it's being considered further now. Is our standard marker genes, which can be assessed from from gene expression studies. And these are various genes that differential gene expression with exposure to and then the P effector measure can be applied.
Hello.
So there are various ones, and this has resulted in, as far as TCD goes, TCD has been able to, the modeling of TCD shows that there's one pathway. Its pathway involves several genes that are known. However, as to their relation to cell compliance, uh, uh, has not been previously. These are the ones that are involved are thyroid genes, thyroid-related genes, and um, the deiodinase enzyme itself that is implicated in this regulation. Um, in initial pathway, so therefore this pathway becomes a high affinity. It's been deemed a high affinity thyroxine pathway with increased as the thyroid stimulating hormone gene expression is increased, and uh, 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 um, um, it's predicted to be increased in this pathway, and therefore it's a lesser contraction, lesser expansion gene expression pathway, and this is consistent with what is known and as recently and also recently been reviewed as to with more recent research as to the excuse me building on previous research where actual measurements have shown that there is a reduction in compliance. Reduction in compliance, decrease in compliance in cancer cells, and uh, uh, the pressure here, the mechanism is further determined to involve the thyroid receptor alpha and intracellular deiodinase, and for cell excluded molecules, the cell membrane deiodinase. Um, so the second compliance pathway is TCD-like PCB-associated gene expression pathway that is identified based on these similar PCBs that are less toxic in terms of dioxin, dioxin toxicity. And this is a continuation of a continuation of the initial pathway in the spectrum. And then the final pathway, the second pathway, the latter pathway, is the non-TCD-like PCB-associated gene expression pathway, and this involves certain standard marker genes that are overexpressed in based on which other genes with this with the measure can be interposed and matched as to genes that have not previously been known to be. And there's much literature now on human genes as to the genes that are co-expressed, and this further delineates these relationships. Um, and this is the ARNT pathway. So by this in this research, it has been possible to separate the aerial hydrocarbon receptor pathway from the ARNT pathway and androsten receptor pathway, which is this is the latter pathway, the third pathway, in which the thyroid receptor B and beta and deiodinase 1 and 2 one are implicated along with estrogen receptor beta within this pathway somewhere. This is itself an more towards apoptosis. Just before this pathway, it would be the previous pathway, which is more consistent with mitoses, and so this would be more towards the telophase interphase, where there is a direction to apoptosis also. Um, but so this is this pathway. Now, in terms of the implications, well, cell compliance has been measured before, and it's shown that it is increasing near transformation. But now it is possible to predict based on gene expression and cells express different genes as to open and close motifs. So therefore, any differential expression or any can be applied the top-ranking genes in both categories, negative and positively, to determine where a cell may be within the spectrum, and this can be applied to, of course, personalized therapy, um, where where this can be where for example can in treatment eventually, hopefully, in a translational treatment regimen where response to therapy can be as to where the cell belongs, uh, as far as translational implications, in addition to the basic science knowledge, basic science knowledge of basic science, um, um, addition to the literature on the mechanisms of regulation of cell compliance with the and this being the deiodinase system. Uh, and uh, and there's further work ahead. Um, so conclusion wise, toxicants result in neotransformation or within the spectrum of neotransformation apoptosis. Cell differentiation occurs, also de-differentiation occurs, and this um, can be falls within a spectrum, and it is since there's genomic mutational interactions that are that result in the resistance potentially effective chemotherapy. This is a is a this is applicable, translationally applicable, the research that the research on uh, 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 with uh, with, for example, serum RNA markers or tissue markers. Um, I appreciate the input of previous mentors and supervisors, which are listed here, and also of the organizing committee here, and the opportunity to present this for day one here. At the conference, and to the session chair, Dr. Dinesh Bharani. Um, if there are any questions, I can answer them here or via email or